This video is about the paper called Self-supervised multi-modal alignment for whole-body medical imaging. Let us look at the key points that are mentioned in the abstract. First of all, this is a self-supervised learning paper, and it is in the medical imaging domain. It is used for the case where there are two modalities available, and these modalities are basically Dixon technique magnetic resonance or MR scans and dual energy X-ray absorptiometry DXA scans. They have three main contributions. One is that their approach is a multimodal image matching contrastive framework with the objective of basically matching different modality scans of the same subject with high accuracy. This means that they basically use this as a pretext task. Two, using the correspondences learned during this contrastive training step, they basically, without any adaptation, use them to perform automatic cross-modal scan registration in a completely unsupervised manner. And they also use these registrations to transfer segmentation from DXA scans to MR scans, and that helps with segmenting anatomical regions without requiring the ground truth MR examples. Their codes also are going to be available at this GitHub account. So starting the introduction, they start with talking about why annotated datasets are hard to get, especially in the medical tasks. One is because using patient data requires ethical clearance, and two is because generating ground truth labels may require expertise from clinicians whose time is limited and expensive. And three, that clinical datasets are highly imbalanced mostly, and the number of negative examples are usually larger than the positive examples. Therefore, acquiring large datasets is very expensive, time-consuming, and frequently infeasible. And as usual, this lays the ground as for the motivation of going with the self-supervised pre-training in order to reduce the need to rely on accurate human annotated labels. So continuing the introduction, they mentioned that they have developed a self-supervised learning approach for cases where pairs of different modality images correspond to the same subject. We still have yet to know what exactly is subject here. And they introduce a novel pre-training task, the pretext task of basically model matching together different modality scans that show the same subject. And basically they want to learn a joint embedding space, and they map both of these different modalities of observations of their subject entity to this new semantic representation, and then they basically take it from there. Now, in itself, they mentioned that this pre-training task is very important because embeddings obtained from this trained network basically allow to check if two different scans show the same subject in large anonymized datasets. And that basically is done by verifying that their embeddings are a good match. And it also helps defining a notion of similarity between the scans. But they mentioned that this task on its own is not their focus of their study, but it is the semantic spatial representations that are useful for the downstream tasks. And they have brought the organization of the paper here. So the related works start with the obvious observation that self-supervised representation learning is a very active area of research at the moment, and that major successes have been reported in image classification domain and natural language processing, and that due to the current circumstances regarding the medical imaging, this is a promising approach probably in the medical domain as well, and some of the previous works have been in this area, an instance being improving the automated diagnosis of intravertebral disc degeneration and common segmentation tasks, and also the work in 32 that shows improving the performance in brain tumor segmentation using multiple MR sequences in a self-supervised fashion. And then they mention examples such as many examples in the computer vision domain that they combine audio and visual streams as a multimodal approach, and show that this helps in making improvements to the different methodologies performances on different tasks, such as these examples. And therefore, they justify that multimodal approach is an interesting research area. Then going to the dataset information, they mentioned that their dataset is obtained from the UK Biobank, which is a large open access medical domain of medical data. Basically, that is taken from over 500,000 volunteer participants. They focus on two whole body imaging modalities that are collected by the Biobank. The first one is the 1.T 6-minute dual echo Dixon protocol magnetic resonance or MR scans that they basically show regions from approximately the neck to the knees of the participants. And the second one is the dual energy X-ray absorptiometry or DXA scans showing the entire body. And in total, they mention that they have, in this data set, they have 41,211 DXA scans and 44,830 MR scans from unique participants. One thing to mention here is that the subject here is basically the patient, most probably. Now, they are looking for same subject multi-sequence MR and DXA scans, and they find 20,360 of these pairs, which they go on in a splitting in 80-10-10 splits. So the modalities are, again, the fat-only and water-only sequences of Dixon MR scans and the tissue and bone images from DXA scans. They synthesize 2D coronal images from 3D MR scans by finding mid-spinal coronal slice at each actual scan line using the method described in one of the references. And as other pre-processing, so all of the scans are resampled to be isotropic, and they are cropped to consistent size of 501 to 224 for MR scans and 800 to 300 for DXA scans. And they mentioned that these dimensions are important because that they basically mean an equal pixel spacing of 2.2 millimeter in both of the modalities. And another important detail is that both of the scans are required with subjects in a supine position, and there can be arm or leg movements between the scans. The first figure that we will shortly see is figure 1, and it basically shows examples from both of these modalities. The objective is to show that the difference in tissue types that are visible in these two different modalities mention that there are information in each one of them that cannot be found easily in the other. And another thing is that these scans are not aligned, which means another source of noise with variation in subject position, pose, and rotation. And this is the example here with the guess who problem or their pretext task. To obtain the similarity, they basically first note that both of the images from these two modalities, namely the DXA and MRI scans, are coronal views, and the subject rotations relative to the scanner are very small. So they define a measure of similarity based on the 2D convolution of the MRI's spatial feature maps over the DXA's spatial feature map, and the maximum value of resulting correlation map provides a similarity score between the two. They will then use this similarity score with noise contrastive estimation over a batch of n randomly sampled matching pairs, such that if Mij represents the similarity between the ith DXA and jth MRI, I equal j would basically be a matching pair, and I not equal to j is a non-matching pair, and tau is going to be some temperature parameter. Based on the noise contrastive estimation, they have this loss for the kth matching pair. So here is an overview of their model. They have the MRI scan here and the DXA scan here. After obtaining the spatial embeddings by passing through these parts, they use the MRI scans, normalize the MRI spatial embeddings, and they convolve the feature maps and obtain this heat map here, in which the peak response gives the scan gives the value for similarity, and then it can be used in the contrastive loss for the noise contrastive estimation, and the peak location can help in obtaining the registration transform. The networks are trained with a batch size of 10, Adam optimizer, learning rate of 1e minus 5, and beta of 0.9 and 0.999. They have done a study on the temperature and chose this value as well. The spatial embeddings are 128 dimensional, and each model took 3 days to train on a 24 GB Tesla P40 GPU. To evaluate the ability of the system, they have two mainly quality controls. One task is to retrieve the matching opposite modality scan for a given query scan based on the similarity, and two, to verify if a scan pair is matching or not. For the second task, they have a threshold for the similarity scores, and they use that to obtain this binary decision. The ROC curves and the table here also shows a good performance. And in this discussion part section, they mentioned that this task on its own is also very important. For example, they mentioned that one could select a query ROI in a scan, perhaps containing an unusual pathology, calculate its spatial embedding, and then find similar examples across a very large dataset. They then talk about the unsupervised registration of multimodal scans problem, and they mention that it is non-trivial, that there have been works on it that have not been super effective, and they explored this idea further and developed 3 methods for estimating rigid registrations between the modalities, and each method is assessed by measuring the L2 distance error when transforming anatomical key points from the MRIs to the DXA scans. For these registration, the first method they propose is dense correspondences, meaning that the contrastive framework attains to align dense spatial feature maps before comparing them. This is done by what we discussed, basically measuring the point of maximum response as the displacement between the images, and the rotation between the scans is also found by rotating the MRI scan across a small range of angles and then convolving the feature maps and basically recording the angle leading to the greatest alignment. The other method is the salient point matching, which is a lot less computationally expensive, especially when done on a CPU, and they only use a few salient points between the feature maps, and they basically use Lowe's second nearest neighbor ratio test and ransack estimation in order to obtain the registration. And the third method is the refinement regressor. This part basically takes the almost aligned feature maps predicted by the aforementioned methods and then outputs a small refinement transformation. This is because of the limitation that the resolution of the feature maps imply, which is the 8 times downsample of the 2.2mm pixel spacing of the original image. They take the salient point matching method's outputs and slightly misalign them with a randomly sampled rotation and translation, and then they train a network to regress this random transformation. And they train this on 50 aligned pairs from the previous salient point matching method. The rest of the paper is more details on these results and basically explaining that the registration and subject matching are done perfectly. So this is another example of the registration method's results, and you can see that it seems that everything is very accurate. The results on the key point transfer error also indicate that that these 3 proposed methods are very effective. And another use of this registration technique is basically that it helps with the transferring of the segmentations between significantly different modalities, and thus making the task of manually labeling these images a lot less cumbersome to carry out. Thank you for watching this video.